but we're doing organotypic human brain slices. So what this means is that normally when we get brain tissue, we can keep it alive for probably around about 24 hours. There's a few tricks that you can use to kind of push it out a little bit further, but if you want to keep it alive for, say, 7 days or 10 days, the kind of periods that you need really to get good transfection, then you have to use organotypic approaches.
Fellow Homo sapiens, welcome back to Epilepsy Sparks Insights. Now, this is a bit crazy to me that I rarely hear about people speaking of brain tumours and epilepsy in the same sentence, because seizures and epilepsy are a really common side effect of brain tumours. Now, hear more today from the brilliant Mark Cunningham of the Cunningham Lab at Trinity College Dublin in Ireland, who shares with us his exciting research into brain tumour-related epilepsy. A quick one, please don't forget to like, comment and subscribe. And now onto our star of the week, Mark Cunningham.
And I'm the Ellen Mason Bates Professor of Neurophysiology of Epilepsy, and I'm based in the discipline of physiology in the School of Medicine at Trinity College Dublin. And I did my PhD, a long time ago now, in Bristol with Roland Jones. I was looking at anti-seizure medications and how they altered synaptic transmission in the brain. And then I went off and spent some time looking at neuronal oscillations, brain waves, and kind of got back into epilepsy through that, looking at high-frequency oscillations. And I've done some work with Roger Traub, who's a computational modeler, looking at the mechanisms behind those. And then I went to Newcastle and I spent 13 years in Newcastle and very exciting time in Newcastle. I set up a platform to do recordings in human resected brain material from patients with refractory epilepsy. And I suppose that's kind of informed a big part of my research since then, is trying to use human material from epilepsy patients, combining that with electrophysiological techniques, pharmacological techniques, to try and understand how networks in the brain generate certain types of epileptiform activity. And then if we can get a better understanding of that, can we come up with better ways of treating the condition, and in particular the sort of the seizure activity associated with the condition.
And do you focus on specific types of epilepsy?
So a big focus of the lab at the minute is brain tumor-related epilepsy. Getting a tumor is brain tumor is a pretty significant diagnosis for a lot of people. And a lot of patients who have a brain tumor will usually, one of their presenting symptoms may be a seizure.
Mm-hmm.
So then they go and get a brain scan, and of course they'll get the very upsetting news that they have a tumor, but also a significant proportion, somewhere around about 30 to 40%, will also have seizures and will develop epilepsy. And so there is a condition now called brain tumor-related epilepsy. And so we're very interested in that. We're very interested in understanding the mechanisms that underlie that. We're particularly interested in a neurotransmitter called glutamate, which, as you know, excites neurons. And what seems to be occurring in these brain tumors is the tumors like to produce glutamate. They love to produce glutamate. And the tumors are, they're it's a pretty insidious mechanism in that they actually use the glutamate to not only to promote their growth, but also to allow them to kind of move out and occupy new space. So they produce lots of glutamate, and the glutamate gets to such high levels that it will kill neurons. And the glutamate also feeds back onto the tumor cells and makes them grow and move and occupy the space where the neurons used to be, the brain cells used to be. Now, of course, too much glutamate's also going to excite neurons, and it's going to cause them to generate seizure activity. And every time you have seizure activity, you also get more glutamate. So it's a really sort of insidious kind of feedback cycle. So the kind of tumor has hijacked the glutamate system, to kind of promote its own growth, but also to promote seizure generation as well.
What you're looking at is it the activity of the brain whilst the tumour resides, or is it afterwards, or is it both?
So it's probably it's a kind of, I mean, how we study it at the minute is we again work with neurosurgeons here in Dublin at Beaumont Hospital. Some of these patients will have their tumor removed, and there'll be a part of the surrounding brain tissue that's been infiltrated with the tumor cells. We will get that. So I often refer to this as a kind of badlands, where you kind of have these kind of nasty infiltrating glioma cells rubbing up against kind of, brain cells.
Yeah.
And it's that interaction. And what we see in those samples is we see seizure activity because there's probably too much glutamate, and that's exciting these neurons. So we're trying to work on a way of potentially treating that at the minute. We're funded by Science Foundation Ireland, and this work takes place as part of the Future Neuro Centre, which is an SFI funded centre. And we're actually trying to use a novel gene therapy approach. So we're trying to target the brain cells with a particular channel that's activated by glutamate. So because there's too much glutamate, when the glutamate acts on this channel that we've expressed in the brain cells, it will actually turn off those brain cells. It will quieten them down. And so we're hoping that might be a novel way of treating seizures that are seen in brain tumor-related epilepsy.
This sounds like, as well, as if you quieten down those cells, then you're effectively protecting the undamaged so far cells as well. And how far along are you in this specific part of your work, and where in the process of the whole study?
So when I arrived in Dublin 5 years ago, the first thing to do was to get ethics set up, because obviously you have to have ethics in place to do the human element of this work. And I was in the process of doing that, and then COVID came along.
Oh, yay! Great timing.
Yeah, so COVID kind of slowed things down a bit, because, of course, a lot of ethic committees were very focused on COVID-related projects. But we're up and running now. We've been going for about a year, so the human recordings are going. A big part of this project is also an animal model. So we're using an animal model in which we express tumor cells, and then they grow a brain tumor. And for that part of the project, what we're doing is, so the human bit is ex vivo, in vitro brain slices. The animal part is in vivo. We're actually doing EEG recordings, and it'll be like video telemetry. So these mice will have a wireless recording system, electrodes. Will record the seizure activity as the tumor grows, and we'll be video monitoring them so we can, capture the seizures. And then the idea is that we would run a sort of clinical trial almost, where we would compare animals that have had the gene therapy approach versus untreated animals. And then the ultimate goal is to translate that into the human brain slices. Now, that's a little bit more tricky, because with the gene therapy, you need an amount of time so that the cells actually start to express the channel that the gene is expressing in. But we're doing organotypic human brain slices. So what this means is that normally when we get brain tissue, we can keep it alive for probably around about 24 hours. There's a few tricks that you can use to kind of push it out a little bit further. But if you want to keep it alive for, say, 7 days or 10 days, the kind of periods that you need really to get good transfection, then you have to use organotypic approaches. So that's where you take the slices, you keep them in a very special brain juice, as I call it, and you stick them in a special incubator, and they cook away for you, and you can keep them alive for sort of weeks, and that gives yourself enough time to get the viral vector in there and express the genes in your cells.
Oh, goodness. And so what do you reckon, when does this project officially end? And in funding wise and stuff?
Funding wise, it's got another couple of years, another two years. So we're hoping that by the end of the project, we have a very good body of, both preclinical animal work, but also proof of concept in human tissue as well. And I think, then we need to think about sort of probably the next step in terms of, thinking about maybe testing this in a more, in a larger animal model. But the human, one of the things that always really excites me about human tissue work is that you really are that much step closer already in terms of using patient-derived material.
Yeah.
And this is material that's come from the patients. You've got these slices. These slices are generating seizure activity. We're able to record them. We can record them with an individual electrode. We can use multi-electrode arrays. And what we're doing in those tissues is we're not only doing the gene therapy work, but we're also looking at other novel compounds and trying to understand whether there's other approaches that can be used. Again, by targeting particular glutamate receptors. And if you can prove that, if you can demonstrate efficacy, albeit in a reduced model, but albeit a human model, I think that gives a lot of confidence that at least the mechanism of action stands up. Now, of course, when you develop drugs, there's lots of other
Indeed.
problems and issues, but I think it's being able to demonstrate mechanism of action and efficacy, which is very powerful in human pathological tissue.
Often, if it works in a mouse model, that's great, or zebrafish, that's also great. But the fact that you are actually using human tissue and to see the impact, that is pretty amazing.
Like, I don't get in the lab much, but I love kind of getting into the lab for that. It's very, I often say, we're very, very lucky to be doing it. And obviously, we have enormous gratitude to the patients that consent so that we can use, the material in this way. It's such a unique resource. And, I always say to the guys that are working with the lab, you really have to maximize what you get out of this because it's such a valuable resource. And it gives us such unique insights. And there's not many places around the world that are doing it. So I find it extremely gratifying as a scientist to be able to do that kind of work.
And my experience, maybe it's just my experience, but with when it comes to the epilepsies amongst people who've had a brain tumor and the epilepsy is a result of that, it's really not spoken of that much at all. It's a common trigger for epilepsy if you have a tumor of any kind, whether it's cancerous or not, right?
Yeah, and and that's a very, very good point. Again, we think a lot about the gliomas, which are the invasive tumors, but it's also the case for meningiomas, which are the tumors that affect the meninges. In that case, what happens is you get physical pressure down on the brain rather than an invasive process.
Oh yeah, so it squishes it basically, and that
kind of squishes it. Yeah,
the professional term.
But it still will still cause seizures. So yeah, I do think it's changing. There's a number of people who are working in this area now. There's a lot of good groups in the UK and in Italy as well that are very interested in brain tumor-related epilepsy. So it's definitely there. I think the difficulty lies in that it's kind of it's maybe sort of stuck between two stools because it's neurology and it's oncology, and so you have this kind of area. It's neuro-oncology, but I think there's a lot of interesting work now that's happening, and there's some amazing work. For example, that's coming from there's a lab in Stanford, the Monge lab, and some of the work that they're doing is really incredible in this area as well.
It's lovely that you mention these other labs because I think more and more now labs are working together, right? Sharing where possible so as not to faff about too much and do the same thing more than times than is needed, etc.
Yeah. Well, I think when you look at these the work that other labs are doing as well. It's quite inspiring. Sometimes it's a bit like, oh, damn,
They did it first. Oh,
beaten first, but it is great, and I mean, just thinking I've got there's a lab in Paris led by Gilles Huberfeld, who does a lot of work in brain tumor-related epilepsy. And, Gilles and I are kind of great collaborators now, even though I probably, and so he's visited Dublin, and, I'm on a PhD committee for one of his students. So, these things should lead to sort of positive interactions and collaborations. We can actually gain a lot from one another's work.
A huge thanks to Mark for sharing with us his exciting research into epilepsies caused by brain tumours and what the future may hold. Again, if you haven't already, don't forget to like, comment and subscribe, and see you next time.